Testing, 1, 2, 3, blah, blah, blah. Yeah, that works. Okay. Yeah.
Yes, she texted me multiple times. Huh? Yeah, you're welcome. You know how long it took me to make that one video? All right, it took me like 2 hours. We went for everything you see.
It did 2 minutes.
That's my goal. And the haircut. You know? Oh. Alright, I see you later.
Congratulations. Guys, you took some serious backside.
Yeah, we got our backside kicked the first time. Then we did it back.
So yeah, it was 8, 7.
If I give you one of this,
I don't want to be crying. I was upset because I was like, I did all of their shoppers for the first one, for chapter 25. Did all the problems. So I'm gonna nail that question. And then it was kind of weird. It was like it's 1:30. So I was upset because I was like, this is the one that I 100% is gonna get right. And then it was weird. It was like, but we had 2 things that we don't know. I put equations down for all of them. So I probably got first.
That's me. Yeah.
I just put equations down that have the same. I was the last just because I'm like, I don't know, and then I just move on, but this one I sat there like trying to figure it out. Hey, whatever. I haven't heard of like, I know the process in which I can do this, but I'm missing that. Like, there's too many things.
Okay. If you, for people who are not here, everything that is graded, and I spent a lot of time grading this last weekend. Not just grading, but also making sure that Blackboard essentially would yield to my will. So hopefully things are as they should be with Blackboard. but you never know. All right, so the goal today is to finish the kidney and then to get into the reproductive system. We're skipping chapter 19 one more time, which means chapter 19 will not be on the final. We'll pass go, collect $200 and go straight to chapter 20. All right, so the very high concentration of solutes in the medulla of the kidney is created by two things. It's created by the countercurrent amplification. You have these pumps essentially in the ascending limb of the loop of Henle. These pump a lot of solutes into the medulla, and then the water is taken up by the peritubular vessels. That's the first thing that causes this very high concentration of solutes. The second thing that causes the high concentration of solutes is something called urea cycling. Well, urea recycling, excuse me, urea recycling. Now, here's essentially how urea recycling works. So urea is made constantly in the body. It's a byproduct of amino acid or protein, or in some cases, nucleic acid breakdown, and it's dumped into the bloodstream. So this molecule, ammonium, is very toxic to your body. In order to, when you're metabolizing protein, you make a lot of this. You make so much of this, protein metabolism is going to kill you. So how does your body do it? It sends this to the liver, and via the urea cycle, you convert this to urea. And of course, you're going to need carbon dioxide in there. You're also going to need water in there. You put those in there like that, and out comes urea, and that's not balanced, but we're not going to worry about that right now. So the urea cycle makes urea, and urea is the major nitrogenous waste in urine. So in the initial filtrate, the concentration that's made at the Bauman's capsule in the filtrate, the urea concentration is the same as it is in the blood. So there's 100% of the urea filtered in the proximal convoluted tubule. Something interesting happens as it goes through the descending limb of the loop of Henle. First of all, you lose water. So the urea concentration now starts to go up. Not only do you lose water, but the urea concentration out here is really high compared to the urea concentration in here. So now urea starts to get secreted into the filtrate, so that by the time you get to, so the urea starts to get resorbed in the PCT, so that your urea concentration when you get to the loop of Henle is about 50%. So initially it's 100%, what it was in blood. Now it's about 50%. But as you go down the loop of Henle, you lose water. And now urea gets secreted into the filtrate. So by the time you get down here, you're at 110% of your urea concentration. A lot of urea. And then the ascending loop of Henle and the early distal convoluted tubule are completely impermeable to urea. So that 110% urea stays from down here all the way up to the early end of the early DCT. So to review, filtration, all the urea in the blood goes into the filtrate. Filtrate, 100% urea. PCT, you reabsorb half of it because you're going to use it to take up things. It's not charged, but you can use it then to take other things up as well. Then as you lose the water, as you go down the descending loop of Henle, now you're going to start putting urea back into it. So that down here at the bottom, you're at 110% of the urea concentration of the blood. And that stays the case when you get through the ascending loop of Henle and the early distal convoluted tubule, because those tubules are impermeable to urea. Then you're going to go down the late DCT and the collecting tubule. In the collecting tubule, you have a urea transporter called UT1. And UT1 is inducible by antidiuretic hormones. So down this comes, and urea, because ADH is always made at a low level, so some of these cells are going to have some UT1s. That urea now is going to get secreted back into the medulla. So that by the time you excrete this, you're at about 70% of the urea concentration. And if you have high ADH, essentially it might be more like 40% or something like that. But this urea now comes into the medulla. That helps concentrate the medulla. But then some of that urea is going to be secreted back into the loop of Henle. It's going to go up, it's going to get released, it's going to be secreted, it's going to go up, it's going to be released, and secreted, and what? It's recycled. It's recycled. And that recycling of the urea guarantees that you have this high concentration of solutes, in addition to the sodium, potassium, and chloride that you're secreting out into the medulla by those very powerful pumps in the ascending loop of Henle.
So the UT1 is responsible for the excretion of urea from the
Yeah.
collecting duct?
And then the ones responsible for the uptake they're called UT3. UT3. So we have different urea transporters that do their thing. So urea recycling and the counter current amplifier are the two reasons why you have this gradient of solutes as you go down the medulla, so that by the time you get to the renal papilla, you are at about 1400 milliosmoles. So you are 4.5 times more concentrated than you are in blood, because blood is about 300.
How does dehydration affect this process?
Dehydration affects this process in that you have more water being recaptured, but any water that comes out here is taken up by the peritubular vessels. They have lots and lots of aquaporins, they take water right up. So water will not dilute this. And that's why you have those peritubular vessels, essentially, so that any water that gets put out there is taken up right away. But the urea does not. Urea is taken up by the tubule, not the vessels. The vessels here will start to release urea over here, but they don't touch it over here. And so these cells here will start to pump urea into the filtrate here, that then gets released over here and then gets recycled. That's called urea recycling. And urea recycling is a really important part of the concentration that of the solutes that you maintaining that gradient of concentration of solutes that you see in the medulla. Okay, questions? Not the simplest thing to understand. I told you the kidney was complicated, and I wasn't lying, as you saw. Would I lie to you? Well, maybe. All right, we talked about ADH, talked about diabetes insipidus, talked about aldosterone, we talked about renin-angiotensin, talked about, okay, we stopped, no, we got here. Yeah, okay. Hyperkalemia. Hyperkalemia is too much potassium. Hyperkalemia is going to take the T wave, and it's going to cause the T wave to get a little higher and a little higher and a little higher, a little higher, a little higher, a little higher. So the height of the T wave can tell you how bad the hyperkalemia is. So to remember this, you get a tidal T wave. The T wave looks like a tidal wave. You get a tidal T wave if it's hyperkalemia. Whereas with hypokalemia, O stands for Oreo. You're watching the birdie, essentially. You get the U wave. That's how you tell the difference between hypokalemia and hyperkalemia. And we already talked about essentially, as we went through this already. Okay, bicarbonate. Here's where we stopped. So one of the most important buffer systems in your body is a bicarbonate system. So bicarbonate ends up being an exceedingly important buffer in your blood, and it behooves your body to not waste it. So in the proximal convoluted tubule, you have a very sophisticated system for recapturing bicarbonate, and it goes like this. So in the PCT, you have a sodium hydrogen ion antiporter, and you have two different isozymes of carbonic anhydrase, because carbonic anhydrase is going to catalyze the conversion of carbon dioxide and water into carbonic acid. That reaction, remember, depending on mass action, can also go the other way. And if you have it so that you get rid of this often, this reaction will tend to go in this direction, where you take carbonic acid and converting it from acid to water. If, however, carbonic acid is going to hydrogen ion and bicarbonate, then the reaction will tend to be pushed this way. What do we call that in chemistry? The French dude, the dead French dude? Le Châtelet, Monsieur Le Châtelet's principle. So one of those isozymes of the California enzyme, carbonic anhydrase, is adhered to the outer membrane, the apical outer membrane. The other is cytosolic. So this hydrogen ion that's given up by the PCT epithelial tubule cell, combines with bicarbonate to make carbonic acid, which is then converted to carbon dioxide and water. Both of these readily pass through the membrane into the cell, where a second isozyme that is cytosolic converts it into carbonic acid. That carbonic acid just joins the hydrogen ion and bicarbonate. The hydrogen ion then becomes the substrate for the sodium hydrogen ion antiporter. So it essentially gets recycled. The bicarbonate is then exchanged for chloride. You have a bicarbonate chloride antiporter in the basolateral surface of that PCT cell, tubule cell. Chloride comes in, bicarbonate goes out, and bicarbonate goes back out to the blood where it is needed so badly to be a buffer. So this particular system essentially makes sure that we don't lose that bicarbonate in our blood. And this simply explains what goes there. And this works because you have two different isozymes of carbonic anhydrase. One will catalyze reaction that goes in one direction outside of the filtrate. The other inside the cell, it catalyzes reaction going in the opposite direction, because the mass action is pulling it this way, because this disjoining is occurring because you're getting rid of both bicarbonate and the hydrogen ion. That's why the reaction is pulled in this direction inside the cell. Whereas outside the cell, you're pulling the reaction in the opposite direction, because these are being removed because they're being taken up by the cell so quickly. So it's through that two different isozymes of the same enzyme that drives it, and the fact that mass action drives that reaction into different directions. And this simply shows you, this is a drawing I did using actually ChemDraw. Remember that these cells in the PCT, they're held together by what are called tight junctions. Those tight junctions ensure that the cells are pressed very, very closely together. You get very little leakage through these cells, and therefore essentially those tubules are pretty tightly pressed together. They also sit on a nice basement membrane, and then you exchange the carbonate here and then you have your sodium potassium ETPase to make sure that the sodium that comes in is going to. That's right. When you have acidosis, too much hydrogen ion in the blood, just about all that hydrogen ion that goes into the filtrate will be consumed essentially, grabbing the filtered bicarbonate. But during alkalosis, there are fewer hydrogen ions that are available. So less bicarbonate gets resorbed, so you tend to get more bicarbonate release. Now, because of the buffers that end up in the urine, the acidity of the urine is usually not going to fall below 4.5. That's usually because of the phosphate and the ammonium. The times when pH of the urine may change is if you have an infection of the urinary tract with organisms that make an enzyme called urease. So members of the genus Proteus, a common cause of urinary tract infections. Takes urea and converts it into ammonium and carbon dioxide and water. This ammonium is going to raise the pH initially, but it's also going to give up a hydrogen ion, and it's going to acidify actually the urine. That may drive it below 4.5. And then you may have certain salts falling out in solution. And you can get what are called struvite stones if this happens. Struvite stones tend to branch throughout the calyces. They're very, very hard to get rid of. You usually have to use lithotripsy to get rid of them. And they are immensely painful because they're oddly shaped. They can be broken up, they're fragile, but they are nasty, 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 nasty. Now, what if your kidneys aren't working hard enough, fast enough for you to get rid of fluid? Say you have some sort of, you have heart failure or edema because of some sort of blood condition or something like that. You can use medications called diuretics. The diuretics will work in the kidneys to increase the amount of urine that you release. You will tend to make more urine that is less dilute. There are several different groups of diuretics that work at different places in the nephron. The ones I'm going to focus on will be the thiazide diuretics. They're going to inhibit that sodium chloride channel transporter, that we saw in the early DCT. And then loop diuretics are going to inhibit the very powerful salt pump in the thick ascending limb of the loop of Henle. We also have the aldosterone antagonists that will work in the collecting duct, also in the late loop of Henle as well. And then there are also the bicarbonate, the carbonic anhydrase inhibitors that will work in the PCT. So thiazide diuretics have this common thiazide structure. These are pretty commonly used. These are dirt cheap. That's one of the reasons they get used quite a bit. They're going to inhibit that sodium chloride transporter in the early PCT. You keep more solutes in the filtrate, therefore you keep more water. So you release more water, you release more salts. Carbonic anhydrase inhibitors are not used that much. Acetazolamide is used for high altitude sickness. But it's not, they're not used as diuretics because you tend to get rid of more bicarbonate. That can tend to cause severe alkalosis. So they're not used that much, but they will cause you to get rid of more bicarbonate because they inhibit carbonic anhydrase. They therefore will increase the amount of urine that you release. And then the loop diuretics, furosemide is one. Furosemide is the drug, trade name is Lasix. It lasts 6 hours, and they will inhibit salt transport from the ascending limb of the loop of Henle. And then your osmotic diuretics, they don't get used very much for diuresis. They get used more to crack the blood-brain barrier to let drugs into the brain. They're very good for that. So they use for certain anti-cancer drugs that you want to get into the brain. You give them with mannitol and then they get into the brain. They're pretty nice for that. They can also be used if you have increased intracranial pressure. They can be used for that as well. So mannitol, glycerol, drugs like that. Our osmotic diuretics, potassium-sparing diuretics include your aldosterone antagonists, things like spironolactone and other drugs like that. Eplerenone, they will stimulate sodium reabsorption and potassium secretion. These are some of the weaker diuretics, but they're good if you combine those with other diuretics and you don't want someone to have hypokalemia. Acute renal failure, that means your kidneys stop working. You'll see this in cases of shock. You might see this in cases of someone who's lost a lot of blood because of a hemorrhage or accident or whatever. This inability of the kidneys to regulate volume, pH, electrolytes, excrete waste usually may require short-term hemodialysis or something like that. Ischemia of the kidneys due to certain drugs. Inflammation of the kidney due to glomerulonephritis. That's usually an autoimmune condition. That can destroy the glomeruli, and these can lead to chronic renal failure. Certain autoimmune diseases, systemic lupus erythematosus, so can acute glomerulonephritis that's caused by streptococcal A infections. This is usually due to antibody antigen complexes that get stuck in the glomerulus. That's a type of type 3 immediate type hypersensitivity. Same thing with systemic lupus erythematosus. These may eventually lead to inflammation of the kidney, and it may cause eventually renal insufficiency. Under conditions of chronic renal insufficiency, dialysis is necessary, either peritoneal dialysis or hemodialysis. And if you want to get away from that, you need a kidney transplant. Very good. Any questions about the kidney before we move on to something perhaps a bit more interesting?
Well, if you end up donating one of your kidneys to Spiros, you can still survive it.
Yep. The other kidney actually enlarges. Take over. Yeah. And your kidney filtration rates are usually fine. It's too bad kidneys aren't like the liver where you could take part of one and it regenerates, but no, kidney doesn't work that way. Sorry. Very good. Let's now take a look at the reproductive system. So we are going to buck normal convention, and we're going to do the male reproductive system first. We should usually go ladies first because that's what polite people do. But one, I'm not polite. Two, we're going to do the male system first because it's simpler. Then we'll do the female system, which is a right bit more complicated. So we're getting the less complicated one out of the way, shall we say. So the male reproductive system consists of the gonads. Those are the testes. That's the site of sperm production. And then you have accessory organs glands usually that will provide secretions that go into the sperm as the sperm make their sojourn from the seminiferous tubules to the epididymis to the vas deferens into the urethra and out to the outside world. That set of secretions that the spermatozoa find themselves in is called semen. Semen consists of sperm and a whole lot of other things that are made by these accessory organs. So the testes consist of a set of very highly coiled tubules called seminiferous tubules. These are organized into lobes. Those twisted tubules all are going to converge at this network of connecting tubules. That's called the reed testes. And the reed testes then will be drained by the epididymis. Well, these are the efferent ductules that drain this, that then go into the coiled epididymis. From the coiled epididymis, you then get into the vas deferens. So sperm are born here. They then go that way. They get stored here in the epididymis. And then the rest is largely a delivery system, if you will. When you look inside the testes, you have two compartments. Inside the seminiferous tubule, that's one compartment. Then you have the cells outside the seminiferous tubules. Those are interstitial cells. Part of these interstitial tissue contain the so-called Leydig cells. Leydig cells make testosterone. The testosterone is necessary for spermatogenesis. So even though these two compartments are anatomically separated, they talk to each other. This is a dog testis, but structurally, it has a lot of similarities to a human testis. You can see the different lobes, essentially, of the seminiferous tubule. You can also see that they are drained by the reed testis, and then they will be drained by the efferent ductules that then go to the epididymis. You see the coiled epididymis up there. Notice the testis is surrounded by a couple of coverings called tunics. There is a tunica albuginea. It's albuginea because it's white. If you peel the layers off the testis, you have an outer tunica vaginalis, and then the tunica albuginea. Look at the tunica albuginea, you can see it's got a lot of smooth muscle in it. If you look at the compartments up close and personally, we can see the seminiferous tubules and then the tissue in between, the interstitial tissue. So here is your seminiferous tubule. There's a seminiferous epithelium. Your Leydig cells out here, those are mostly mesenchymal. And we'll take a little closer look at these cells here because they're kind of interesting. So the outermost cells are the stem cell population. Those are called spermatogonia. Once they start dividing, they will divide more or less through the life of the guy. They divide, they displace a cell towards the lumen. The cell that stays attached to the basement membrane, that cell stays a stem cell. The primary spermatocyte, there's a so-called A1 and A2 spermatocyte. The A1 spermatocyte still has the ability to divide by mitosis. This division, by the way, is mitotic. It's not meiotic yet. The A2 spermatogonia, which is displaced towards the lumen, that cell loses the ability to divide mitotically, and it starts to undergo meiosis. So as the cells go further away from the basement membrane toward the lumen, they become more mature. They also enter meiosis, and as they get towards the lumen, they get further progressed in their meiosis. So the primary spermatocyte divides, becomes a secondary spermatocyte, And then that secondary spermatocyte divides and becomes a spermatid. The spermatid will eventually undergo spermiogenesis. Fairly complicated procedure. And spermiogenesis is the production of spermatozoa from the spermatid. You look at this cell, it's kind of round and nondescript. If you look at this cell, it now has a bullet-shaped nucleus. There's a vesicle that's going to be at the apical end, called the acrosome, and then you're starting to grow a flagellum out this way so that that cell can become motile. All of these stages are part of spermiogenesis. They occur essentially when the spermatid gets here, because the spermatid is now going to take its nuclei, its DNA, and it's going to wrap it down really, really tightly. So that it now, you essentially, any of you ever bought a mattress? Remember when you open the box, the mattress is like a jack in the box. Then you try to fold it in the back of the box, you can't. You just can't. That's what the spermatid does to its DNA. It does what happened to that mattress inside that box. So the histones are replaced with proteins called protamines. Protamines are really positively charged. You take that DNA and they wrap it tight, and gene expression gets completely shut off because the DNA is wrapped up so tightly. So one of the gonadotropins, follicle stimulating hormone. There are receptors for follicle stimulating hormone in the Sertoli cells, in the seminiferous tubules. Luteinizing hormone is the second gonadotropin made by the anterior pituitary. But receptors for it are found in the interstitium. The Leydig cells receive luteinizing hormone and they make testosterone. And then testosterone 1 feedback inhibits the production of gonadotropins. 2, it stimulates the seminiferous tubules to make spermatozoa. And the seminiferous tubules, in response to FSH, make inhibin. Inhibin will feedback regulate follicle stimulating hormone. Inhibin is also made by the ovaries in response to follicle stimulating hormone, keeping follicle stimulating hormone within a certain range during this part of the life. And then testosterone indirectly inhibits the secretion of luteinizing hormone. I say indirectly because, as it turns out, testosterone gets to the brain, and even in guys, it's converted to estrogen. And estrogen is what actually shuts down luteinizing hormone production by the anterior pituitary. If you put mutations into the estrogen receptor in male mice, those mice are sterile. So estrogen signaling is necessary for male fertility. What it does precisely is not exactly known, but it's important. And so the testosterone, as you can see here, is subject to an enzyme called a aromatase. It's a 5-alpha aromatase. It's a one-step deal, and it converts testosterone into estradiol, which is a estrogen. So in males, gonadotropin production tends to decline very gradually. It doesn't do the menopause thing like it does in women. And the major androgen that's made by the male testes is testosterone. And then there is a 5-alpha reduced derivative called dihydrotestosterone, or DHT. DHT is responsible for many of the physical changes associated with puberty in males, secondary sexual characteristics. So here's what androgens do in males. They're important for spermatogenesis. So at puberty, they're important. They're not important for initiating meiosis. FSH does that. But for maintenance of spermatogenesis, testosterone is essential. Follicle stimulating hormone is necessary to start spermatogenesis. It's not important for the maintenance of it. Testosterone is. It's important for sex determination during growth and development, during fetal stages. It's important for secondary sex characteristics. The progression through the so-called Tanner stages of sexual development. And then anabolic effects. Proteins, muscle growth, growth of bones, growth of the larynx, so the voice lowers, and erythropoiesis, and so on and so forth. Because of this, androgens and other synthetic androgens are called anabolic steroids. And these are the drugs that get abused sometimes. They don't have a lot of medical uses, they have a few. If you have someone who's wasting away because of either cancer or HIV or something like that, anabolic drugs are available. They are used, but they're used sparingly because they're very powerful. These will also stimulate maturation and growth of the accessory sex steroid, sex hormones of sex organs, prostate, seminal vesicles, copper spine, things like that. Lowers the voice because of the growth of the larynx and promotes hemoglobin synthesis as well. So back to these estrogen receptors, turns out the Leydig cells and also the Sertoli cells inside the seminiferous tubules, they have estrogen receptors as well. And those estrogen receptors are essential for male fertility. What precisely they do, not completely sure, but it's clear that testosterone from the Leydig cells gets metabolized by the seminiferous tubules and other molecules. Those other molecules are essential for spermatogenesis. One of those molecules might very well be estrogen. And then the bit about inhibin as well. Remember that the seminiferous tubules are sensitized to testosterone by follicle stimulating hormone. And then follicle stimulating hormone will also sensitize Leydig cells to luteinizing hormone. Leydig hormones also make a few peptide hormones. The role they play in fertility, not terribly clear, but Leydig cells make testosterone, but they make some other stuff too. ACTH, MSH, and beta endorphin. Spermatogenesis. So during early embryonic development, during about the second week of early embryonic development, the embryo makes something that used to be called the yolk sac. It's not called that anymore. It's called the primary umbilical vesicle. Layer of cells from the inside of that primary umbilical vesicle will detach and become motile. They'll start to move. They'll actually move because the gonads aren't present yet. The cells will move up through the GI tract, and then they'll get to about the beginning of the midgut. They'll move through the midgut down into the developing gonad, because the gonad really isn't around until about third to fourth week of development. They will get inside the developing gonad, and if they are XY, they will develop into spermatogonia. If they're XX, they'll develop into eggs. And then the developing gonad will surround them, and the surrounding spermatogonia will then be surrounded essentially by elements of the medullary parts. You get spermatic cords that will essentially envelop them, and they'll develop inside these developing seminiferous tubules. If the embryo is XX, it's the outer layer, the cortex essentially, that will take these nascent oocytes and will surround them, and those will become the follicles. That will then become the ovary. So these germ cells, they'll become the spermatogonia, the stem cell population. So they divide, you make a primary spermatocyte. Again, there's an A1, A2, primary spermatocyte. The A1 can divide a few times, and that's it. The A2 cannot. The A2 primary spermatocyte will then start meiosis. It divides into a secondary spermatocyte. Notice these cells stay connected by cytoplasmic bridges. Those are very important. The second meiotic division then produces spermatids. Spermatids stay connected. They then will start to make a structure called a hatchet. The hatchet is the beginning of the flagellum. They will then start to they'll undergo spermiogenesis. Their nuclei become bullet-shaped with a Golgi apparatus-derived vesicle called an acrosome in front of it. The mitochondria will then make a collar around the midpiece, and then you'll have these large arrays of microtubules that will then be the motility machine for those cells. As we said, the mitotic division causes only one cell to stay the stem cell, the other cell gets displaced towards the lumen. That becomes the primary spermatocyte, the A1. One of these cells will then divide a bit more, become an A2, then undergoes meiosis, the other cell stays the stem cell. So you'll initially have 1,500 or so spermatogonia. Those cells will divide by mitosis. They'll become quite a few more. These cells spend their time essentially embedded in a larger cell. This cell is called the Sertoli cell. The Sertoli cell acts like a nurse cell for the spermatogonia. So the spermatogonia, as you see, surrounded by the Sertoli cell, its products stay ensconced within the Sertoli cell. The Sertoli cell is going to protect and nourish the developing spermatogonia. These cells are going to dump a lot of their cytoplasm. That cytoplasm that they dump is called the residual body. The Sertoli cell is going to gobble that residual body. In order to do that, that needs a signal, and that signal is testosterone. When it gobbles up that residual body, the spermatozoa lose their attachment to each other. You can see they have that nice bullet shape with a nice collar that's full of mitochondria. And at that time, they are cast away into the lumen. And then the seminiferous tubules, they're not motile at this time, but the seminiferous tubule will actually contract and send them to the ductus deferens that then efferent ductules that take them to the epididymis, that then take them to the vas deferens. In the epididymis, they become motile. All right, so we start with a spermatogonium. It has 23 chromosome pairs, 46 chromatids. It divides by mitosis. That cell still has 46 chromatids, 23 chromosome pairs. Now there goes first meiotic division. Now you have 23 chromosomes. They're still paired at this time. Then second meiotic division, you're going to have 23 chromatids at this time in 4 different spermatid cells. They'll then undergo spermiogenesis that become the sperm cells you see there. The Sertoli cell forms a barrier around the developing sperm. They form the so-called blood testis barrier. And that's why the immune system is never sensitized to the reproductive system under normal circumstances. You don't have antibodies against sperm unless you get a vasectomy or unless you are part of the population of people that are called MSM, men who have sex with men. Those gentlemen have, because of the tears in the rectal epithelium, their sperm are exposed to the immune system, and so they have antibodies against sperm, and they will have a very hard time if they want to father kids, fathering kids. So this essentially just recapitulates essentially the Sertoli cell protecting those spermatozoa as they develop and forming the blood-testis barrier. So just a few words about spermiogenesis. It's going to start with the spermatid handing a lot of its excess excuse me, cytoplasm over to the Sertoli cell. That's a residual body. The nucleus becomes smaller and smaller and smaller as the DNA gets wound tighter and tighter and tighter and tighter and tighter. The hatchet then, the hatchet is actually, you can see the hatchet. The hatchet starts to form by this period of time. You can already see the hatchet starting to form. So the structure that will be the nucleation site for the formation of the flagellum, that you can see that quite early. In order for the sperm to mature, you need testosterone. There's a protein in the Sertoli cell, the androgen binding protein, the ADP. That binds and concentrates testosterone inside the Sertoli cell so that the sperm can mature. The spermatocytes, when they migrate from the primary umbilical vesicle down into the gonad, they don't make sperm until puberty. Because at puberty, then you start to make follicle stimulating hormone. That begins the maturation of spermatids into spermatozoa. It's required for initiation, not for maintenance. You need testosterone for that. And at puberty, of course, testosterone starts to be made in spades because you're also making luteinizing hormone that's stimulating Leydig cells to make testosterone. And so this shows you the formation of the residual body. The residual body essentially being phagocytized by the Sertoli cell. The Sertoli cell then essentially dumping the, would you please stop calling me? Where was I? Yeah, grabbing that residual body and casting the sperm into the lumen. So the seminiferous tubules are connected at the root testes. They're then drained by efferent ductules into that twisted epididymis. This is just to give you orientation of where you are. So this is the testes. You have the seminiferous tubules in lobes here. They're drained by efferent ductules into the epididymis. The epididymis is highly coiled. That's then drained by the vas or ductus deferens. That vas or ductus deferens is paired. There's one for each testis. That swings around and behind the prostate and then inserts into the back of it. Where it inserts into the back of it, you have the so-called ampulla. The seminal vesicle will provide its contribution to semen, and that little ductal system there, that's called the ejaculatory duct. This part of the urethra, this is called the prostatic urethra. The ejaculatory duct also will receive contributions from the golf ball sized gland called the prostate. The sperm with the secretions of these two glands will then enter the urethra. There's another gland here that's going to give its contribution of bulbo-urethral gland, also called the Cowper's gland. And then this material, there's also, when this is open, there's a check valve that closes here. So you don't get a mixture of urine with semen. That would be gross. So when this is open, that's shut. When that's open, that is shut. And then it goes through the urethra, which is in the penis here, and then out to the outer world. The epididymis is really tightly coiled. When you looked at it, you might get the idea it's just kind of a twisty sort of tubule. No, it's really highly coiled. If you stretched out the epididymis from each testis, it'd be 5 meters long. Very tightly coiled. So again, that mattress inside it. Yeah. It's then drained. The epididymis is drained. And the epididymis, by the way, is a storage area, and it's here that the spermatozoa become motile. They pass through the epididymis into the vas deferens. Within the vas deferens, the sperm cells get modified. They become more resistant to changes in temperature and pH and other sorts of things. Here is a cross-section of a vas deferens. Notice the vas deferens is mostly muscle. The vas deferens is not an open tubule, it's a contractile tubule. There's lots of smooth muscle, and then looking at the mucosa itself, we see a pseudo-stratified ciliated columnar epithelium. There are basal cells there. Those basal cells are the stem cell population that replace cells as they're sloughed into the vas deferens. From there, you go to the ejaculatory duct. Once that little check valve at the ampulla is open, the seminal vesicle actually contracts. It doesn't just, oh, here it comes. No, it contracts and it squirts its secretions into the fluid. And the main contribution is fructose. That'll be the main energy source for the sperm, because the sperm are going to need an energy source where they're going. There's not going to be a lot of energy. You can see the seminal vesicle there. Here's a cross section of the seminal vesicle again. Tell me what you see right here. What does that look like? Muscle, yeah, muscle, it's contractile. And then you also have an epithelium, that's your secretory epithelium. That looks nice and columnar, like a secretory epithelium should look. And then you secrete it into the lumen there, and then that lumen contracts. And then the prostate adds its bit. The prostatic secretions include citric acid. Citric acid is an extremely important buffer because inside the woman's reproductive tract is acidic. The sperm are going to die if they hit all that acid. So citrate is going to neutralize the acid in the birth canal of the female. And there are also coagulation proteins. These are going to take the sperm and clump them together so that in the hostile environment in which they find themselves, they're clumped together and that helps them survive. They're kind of like, maybe if you've seen the movie 300, how did the Spartans fight? They fought as a phalanx. Shields up, like that. That's what the sperm do. Shields up, that maybe the outside guys get it, but the guys behind them, they also die. Right. Hey, it works. This is a really nice summary of the contents of semen from your book, Table 20-5. This shows you essentially that the volume of ejaculate varies from anywhere from 1.5 mils to 5 mils. And the sperm count also is going to vary. The sperm, if you look at them, not all of them are going to be motile. A percentage of them will be motile, hopefully the majority of them are motile. There'll be some white cells. You can have a cold and have actually more white cells pH of the sperms, you can see will be alkaline, that's due to the citrate. And then the fructose concentration, complements of the seminal vesicle, will be anywhere from 100 to 150, 600 mg per deciliter. So here is you can see the location of the prostate. Here's a picture of the prostate again. Lots and lots of fibromuscular stroma, secretory cells, but also lots of muscle that will help it contract and drive its secretions into the prostatic urethra. These organs are all androgen dependent. Without androgens, they will wither. And we're out of time now, but we'll get to the male sexual response. It's the 3 E's, so it's easy to remember. And we'll cover this on Wednesday and then move on to the ladies. Now, let me just say one thing. Some of the pictures I'm showing you are a little bit graphic, but I am trusting you all to be nice and mature, to come to class and get your Snickers out before you come to class, okay? This is not pornography, it's physiology, right? And Dane, you're married, you have no excuse. How you feeling?
I feel fine. I talked to Holton Health today because I was going to go in and get a second test, but they said that I started showing symptoms like last Monday. So I'm past their isolation period. So
You're on the downside.
Hmm?
You're on the downside of this.
Yeah. So do you want me to come to lab tomorrow or no?
See how you feel? Do you have a home test?
I don't, not
You can go
with
to the
needle.
drugstore and get one.
Okay.
If your home test is negative, I'd say come in.
Okay. Sounds good.
The other option would be to come in and wear a mask.
Okay.
Although if
yeah,
you're
I'll-
negative, I would say a mask is not necessary.
Okay.
So either or, 6 of some, half dozen of the other, up to you.
Okay. Sounds good.
Okay. All right.
Thank you.
Ciao, babe.
Yep, bye.